This video talks about groundwater sampling in general and describes the operation of a passive sampler for groundwater. Groundwater means water that runs in the empty spaces within the soil and bedrock. It does not mean an underground lake, but water present in underground pores. Groundwater starts as rain and surface water that is absorbed in areas where the soil is coarse. The water is purified as it percolates through the soil. Sandy eskers and marginal formations are typical groundwater catchment areas in Finland. Groundwater catchment areas have provided clean water for human societies, but due to industrialization and spreading settlements, their condition has deteriorated, which is why their need for protection has grown. When studying groundwater, you need to know the premises, purpose, and aim of the study. You need to know the background of the groundwater catchment area. What is the structure of the groundwater catchment area? What are the flow directions of the groundwater? Or is the groundwater layered? What polluting activities are there in the groundwater catchment area? For example, are there factories or other risk factors in the area? What substances may have entered the groundwater? How does the substance behave in groundwater? Is the substance lighter than water or heavier? Groundwater wells are essentially pipes drilled into the ground with a water-permeable screen at the bottom. There are various structures and materials of groundwater monitoring wells for different purposes, for example based on the target contaminant. The material of the well should not interfere with the sampling results. For this reason, the wells are often made from PEH plastic. Due to rusting, steel and iron pipes are only suitable for level measurements. Groundwater wells are usually installed so that they run from ground level all the way to the bedrock. A well card for an installed well is always prepared and contains the key information about the well, such as the length of the well and the screen, the coordinates, the type of soil, and the level of groundwater at the time of installation. A locked steel casing is installed around the actual groundwater well. Information about groundwater wells and other groundwater monitoring sites in Finland can be found in environmental databases such as the Groundwater Information System (POVET). Work at a groundwater well always begins by measuring the groundwater level and the depth of the well. The measurements should be taken from the top of the plastic monitoring well, not the top of the steel casing. The measurements are marked in the sampling record. Other information about the well, such as the elevation of the top of the well and the ground level, can be found on the well card. Groundwater is usually pumped up to the surface. There are various types of pumps available for different purposes. Before taking a sample by pumping, the groundwater should be pre-pumped. This is to ensure that the water has not been stagnant in the well and represents the groundwater itself. The pre-pump is done at the target sampling depth. Pre-pumping begins by pumping 8 to 10 liters of water per minute for at least 5 minutes, or until the water turns clear. After the water turns clear, the pumping power can be increased and water is pumped for at least 15 more minutes before taking a sample. The total pumping time is therefore at least 20 minutes, during which time 100 to 300 liters of water is pumped up from below. By then, the water in the well has been replaced several times. Once pre-pumping is done, the actual groundwater sampling can begin. Bottles are usually filled so that several times more water than the volume of the bottle is allowed to flow into the bottle. This rinses the sample bottle during sampling. If the bottles have been pre-processed or chemicals have been added in them beforehand, they are not rinsed. The type of sample bottle depends on how the samples will be analyzed. The bottles should always be filled according to the instructions given by the laboratory. The samples should also remain unchanged until the analysis. For this reason, some samples must be fixated. For example, oxygen samples are chemically precipitated immediately after taking them. Some variables also have to be analyzed with field meters as quickly as possible. For example, groundwater temperature cannot be fixated, so it should always be measured in the field. Other field measurements of the water can also be performed according to plan, such as the pH value and conductivity using the appropriate meters. The field values are recorded in the sampling record. Sensory observations such as the smell of the water should also be marked in the record. Samples can also be collected from groundwater wells using grabs. They can be used, for example, when the water yield of the pipe is low, the sampling depth is too deep for pumping, or water is required from a specific depth or water layer. Grabs are also used when studying volatile compounds. However, grabs only provide small sample volumes, and using them can mix up the water layers in the pipe. Depending on the case, Groundwater samples can also be taken from other places besides monitoring wells. For example, samples can be collected from springs directly into a sample container, and groundwater can be pumped up from wells using a hand pump. Pre-pumping should still be remembered in these sites so that the water comes from the actual aquifer. For example, 2/3 of the water volume of a well should be pre-pumped before sampling to clean the water. Samples can also be taken from the groundwater that fills the trial pits made when investigating contaminated areas. The reliability and representativeness of the results should always be assessed when taking samples from such points. Once the required samples have been taken, they are packed carefully in a cool bag and transported to a laboratory for analysis. You should also remember to send the analysis form with the samples so the laboratory knows what to analyze from the samples. Passive sampling refers to a method where, instead of collecting water samples, the sampler is left at the target site for days or even weeks. The method is based on chemical and physical reactions between the collector and the surrounding water and the contaminants in it. Passive samplers are always optimized for specific groups of substances such as VOC and PAH, compounds or phenols. The project, entitled New Innovative Methods for Groundwater Quality Monitoring, UMO, in the Southeastern Finland University of Applied Sciences, developed an integrative passive sampler for chlorophenols, which is demonstrated in this video. Various types of passive samplers are also commercially available. The collector of the UMO project is based on the ability of silica discs to accumulate contaminants, in this case chlorophenols. 3 discs are placed in the collector, where they provide parallel samples. The collector is lowered to the target sampling depth and kept there for 2 weeks. During this time, it accumulates chlorophenols and does not release them back into the groundwater. After the sampling period, the collector is retrieved and its analysis can begin. After sampling, the collectors are disassembled and their silica discs are placed into decanters, after which the discs are processed to isolate chlorophenols. For quality assurance, a known amount of internal standard is added to the discs to assess the effectiveness of the method. Preprocessing involves several steps, during which chlorophenols are extracted from the discs and processed for analysis. The chlorophenols isolated from the discs are concentrated to a volume of 1 milliliter, after which they can be analyzed by gas chromatography mass spectrometry. The chlorophenols are identified from the chromatograms produced by the device. Chlorophenol standard solutions are used in quantification. The results of the 3 parallel discs are compared and their mean value is given as the combined result. This makes the results more reliable. The results of the passive sampler in the UMO project describe the accumulated contaminants during the sampling period, or in this case, how much chlorophenols there were in the groundwater over 2 weeks. A passive sampler can be helpful when investigating whether chlorophenols are transported in groundwater flow over a longer period of time. Passive samplers are more likely to detect contaminants due to their long sampling time than a single sample drawn by pumping. Due to the increased use of groundwater and human settlement, the need to protect and study groundwater has grown. However, studies are not useful if the results do not represent the actual state of groundwater. Appropriate, high-quality groundwater sampling and the obtained results allow us to assess the state of groundwaters and respond to any problems. Groundwaters are extremely important environments both for humans and for nature, which is why we should take good care of them.